All right, and for the last topic in this module, we'll talk about how we can use molecular data to help us gain information about foraging ecology of different species. So overall, foraging ecology studies the behavior of animals when they're seeking and consuming wild food resources. And this relates to some topics we've talked about in this class previously, because oftentimes nowadays the way that they're doing these types of studies is using metabarcoding, which, remember, is using some type of universal primer to amplify, for instance, all arthropod species. They can often get these samples from poop. That's poop. You can get fecal samples, you can extract DNA, use universal primers, and then do high throughput sequencing to be able to use that data to identify what prey species were there, and that can tell us about the diet of the animal whose poop we sampled. And that often ends up the data is summarized in these taxonomic diversity plots like you actually created when we did the DNA subway lab in this class. So foraging ecology is important for a couple of different reasons. One of the reasons knowing about foraging ecology is important is because it can provide information about how species get the behavior that's associated with food acquisition. So it can provide insight about food acquisition behavior. So this image I'm showing here is only kind of related, but I have to share it because it's related to one of the coolest talks I ever saw when I was a grad student at Scripps. So these are orcas. You can see 3 of them under the water here. The story is not great for the seal, so don't get too attached to the seal. But what I think is super cool is one of the ways that these orcas hunt is they will rush up to an iceberg and they'll create these giant waves, which will then knock the poor seal off of the iceberg. And so this is food acquisition behavior, and they learn this behavior by watching the other whales in their pod. So they learn this by watching the other older orcas. And what I thought was so cool is the person who was explaining this had actually been, I don't remember exactly where it is, but he had been somewhere where there are wild orcas. And he was saying that the baby orcas who don't know what they're doing yet, they can't tell a difference between an iceberg and a small boat. So the babies, while they're trying to learn how to do this behavior, will get confused and come up to a small raft and try to knock the people out of the boat because they don't know what they're doing yet. But I thought that was super cool. So what foraging ecology can give us is information about how species pass on food acquisition behavior to their offspring. And an example of this and how they used molecular ecology data in terms of relatedness to inform this was a study done on black bears, and I think this was in Yosemite, actually. Yes, it was in Yosemite. And so what they did is they had data about bears that would raid campsites and forage there. That's what's shown here. And when they had that data combined with relatedness data about which bears were the moms of different offspring, what they found is there was actually a correlation here between the mothers that were raiding the campsites actually seemed to be teaching this behavior to their offspring, who then were also raiding the campsites. So this relatedness based on genetic data revealed that social learning is one of the ways that black bears are learning to forage on human food. Another thing that foraging ecology can give us information about is just generally the diet of different species. So here in this graphic, again, often this is being done with metabarcoding studies. So in this example with this seal, you can use metabarcoding to figure out what different types of prey, what different types of fish this species is eating. And this is important for many reasons, but it has conservation implications too. And ties into what we talked about in the previous module. If we know what food different species are eating, if that food starts to decline, that might indicate it could be a problem for this species, especially if the species is already threatened or endangered. We want to make sure that we're also protecting whatever food it eats. If it doesn't have food, if that food starts to decline, that could be a problem. So one example of this that's related to the study I referenced in the overview video, but it's actually a different bat diet study. Apparently there have been a lot of these recently. What they did is they used metabarcoding to determine that the common bent-wing bat, which I'm showing right here, contributes to pest control. So again, I thought this was an interesting example because it gives an illustration of how we can use this type of metabarcoding data that we might think is more ecology focused, but it also does have some agricultural human implications as well. So in this table that I'm showing here, this is listing some of the different arthropod pest species that were found to be in the diet of this bat by using this metabarcoding approach. So there in the table here, there's the pest species, the plants, the crops it affects, and then the number of different samples that was found in these different bat samples. Lastly, foraging ecology can also give us information about the different routes that species take to their foraging grounds. So the images I'm showing here are an example based on gray whales, which are another species we can see here off the coast of California during their migrations. So they move these enormous distances across the coast of North America. So up here in the north, in Russia, is where they feed in the summer. So those are their foraging grounds. But the route they take to get there is enormous because what happens is they actually give birth down here in Mexico in these different lagoons. And that's the picture that's shown here. I never made it down there, but I knew several people when I was living in San Diego. You could take trips to these lagoons in Mexico, and the gray whales there are super friendly, and also the babies that have just been born are really curious, and so you can go and get really close, of course observing all guidelines to make sure you're not injuring the whales and whatnot, but you can get really close and have these really, really cool encounters with these gray whales that have just been born. So they're born down here into the winter birthing and mating grounds, then they have to move all the way back up here to get to their feeding grounds. So there's this really long route they have to take, so it's important for us to be aware of that, so we know, for instance, even if they're feeding up here, it's important for their overall biology and conservation for us to preserve not only the birthing and mating grounds way down in Mexico, but also this whole route they have to take to get there. So a similar sort of thing was found in green sea turtles. Excuse me. So this study was done in Australia, and in this case they used mitochondrial DNA to figure out, they were sampling turtles from foraging grounds in the Great Barrier Reef, and they used mitochondrial DNA to determine the rookeries or where those sea turtles were born. So they have distinct genetic data for, I think, 25 different rookeries, they said. So by matching the mitochondrial DNA of the turtles that were foraging in the Great Barrier Reef, they can figure out what rookery that turtle was born in, so where it came from, essentially. And what they found was that some turtles that were eating on the Great Barrier Reef, which is around here-ish, I think. Again, not great with geography. But what they found was that they sampled turtles that were foraging on the Great Barrier Reef, but by matching their mitochondrial DNA with the different potential rookery sources, they found that some of those turtles actually came from 2,000 kilometers away in New Caledonia. So again, this is giving us information that the turtles who are feeding on the Great Barrier Reef actually come from really far away. They travel these huge distances. So if we want to make sure we're conserving that species, we have to conserve its rookery, its foraging ground, and also that whole route that it has to take to get there.